Hi everyone, welcome to our channel NCERT Science. So this video is a continuation of the NCERT Science Class 9 series, and today we will be talking about Chapter 5 in NCERT Science, the fundamental unit of life. So until the previous chapter we were doing chemistry, and this is the first chapter in biology for Class 9. And before we started with the lesson, I just wanted to mention three things. The first thing is that I want to mention that this video will be helpful to those preparing for CBSE exams, NTSE and NEET. Usually the videos I do are helpful for JEE as well, but this is biology, so maybe not so much. Alright, so second thing is that I encourage you to take your own notes. So I've been saying in every video, and I will tell you in this video as well, I took the pictures from the NCERT Science textbook that I found online, and I will be presenting in my own words, but I highly encourage you to take your own notes because that will help you remember a lot better than if you just copy what I'm just saying. Alright, and the third thing is that while I was going through the videos that I already did, I noticed that I was going a little fast. I am doing so because I want to keep the videos short and concise, for which usually best for those preparing last minute before exams and stuff like that. I just want to make it seem like a small crash course video, so I'm trying to keep it under 1 hour, maybe 1 hour 15 minutes. So that's I'm going fast, but if you face any difficulty, I suggest you to go to the YouTube settings and change the speed to 0.75. So that would help you understand, if that will help you understand a little better. Alright, so then with that said, let's start with Chapter 5, the fundamental unit of life. Alright, so this, as I said before, this is the first chapter in biology. Until the previous chapter we did chemistry, so make sure you have everything right, and you make sure you understand everything in chemistry before you start with biology, so that will make things easier for you, so you can be like, okay, I'm done with the first 4 chapters of NCERT Science, let me move on to the 5th one. Alright, okay, so Chapter 5, the fundamental unit of life. So this is a basic overview of the chapter. Spoiler alert, the fundamental unit of life is the cell. That's what you'll get to know in this part of the lesson right there. So it just talks about a scientist who discovered cells for the first time, and then we have an activity here. So as I said in the previous videos, most of the activities that they give here are things that you can do at home, but in chemistry we came across a few experiments in which there were chemicals involved, so that I strongly insist you do it under adult supervision. And this is one such experiment where you need a microscope of sorts. Alright, and then we have a compound microscope, diagram of a compound microscope. When I was doing 9th grade NCERT, we needed to draw this diagram and know how to label everything for the subjective part of the test. For objective part of the test, they just replaced a few of the parts with letters like A, B, C, D, suppose like that, and then maybe E also. And then there was a question that said name the following in order, right? And then there was option A, which said mirror, base, and then microscope and microscopic slide, eyeglass, and course adjustment. And then there were other orders like that. So long story short, you should know how to draw this for the subjective part. You should know how to label this for the subjective and objective parts. Alright, and then so we have a few questions. what do we observe as we look through the lens that I want you to, hopefully if you have a chance to look through a microscope, I want you to look through a microscope in your school or wherever else you have access to a microscope, and then see for yourself, because it's an amazing sight. And then can we see the structures that we are able to see through the microscope on an observation sheet? I mean, did I say see? No, I mean, can we draw? Can we draw the structures that we are able to see? Most of the time, yes, and I hope most of you can. And does it look like figure 5.2 right here? The answer to this would be a definite yes. Alright. So then, moving on to page 2. Okay, we can try preparing temporary mounds of peels of onions of different sizes. What do we observe, and do we see similar structures or different structures? So the answer to this would be similar structures no matter the size. If there is one kind of cell, we always find the similar structure. For example, if you take like a real life example, suppose you have solid X, alright, and this is in a box of this size. So if you look through a microscope, it probably has like particles like this. And if you take the same substance X, but you take a bigger box, it will still contain the same particles. It's just that the number of particles increases. It doesn't in any way, it doesn't change the way the particles are or how the particles look. It's just the number of particles that increase because of the increase in the space or the capacity of the solid. Alright, that's exactly how it is with these mounds of onion peels. Just because we took a different size, it doesn't mean there are different structures. The structures are same, it's just that the number of basic units, aka cells, that is what differs. Okay, so what are these structures? I just answered your question. They're cells. Alright, so these structures look similar to each other. They look similar, so that was the answer to this question. They look similar, and together they form a big structure like an onion bulb, right? So if you have small dots, let's say, together if there are like thousands and thousands, maybe in the end they form a small circle. So this one circle that I drew has many of these individual dots, right? So there's like almost, I don't know, there's so many. Alright, so together they form a big structure called an onion bulb. So smaller basic units make up a bigger whole shape, alright, or a structure. And then we find from this activity that onion bulbs of different sizes have similar small structures visible under a microscope. And the cells of the onion peels will all look the same regardless of the size of the onion they came from. That's exactly what I told you previously, right? So these small structures that we see are the basic building units of the onion bulb and also of every living thing. And these structures are called cells. Not only onions but all organisms that we observe around us are made up of cells. However, there are also single cells that live on their own. So organisms that are made up of 2 or more cells. So let's say number of cells is greater than equal to 2. That would make these organisms multicellular. Alright, and people who cringed looking at this mathematical equation, don't forget you are in class 9. You still have to do all the sciences and math. So I'm just bringing in that math touch. Alright, so they are called multicellular organisms. Organisms, there's an I. Alright, organisms. And those that have the number of cells that are equal to 1, they're called unicellular organisms. Alright, So then we have a little trivia information about cells and who discovered it, how they discovered it, and then we have the cell theory. So then so let's talk about the cell theory. Cell theory has three postulates. The first one, the first two were proposed by Sleeden and Swan. That said, all plants, so let me just do 1 and 2. All plants and animals are made up of cells. Made up of cells, which, that is an ugly W, which are, so which refers to the cells, okay, which are the basic structural and functional units of life. Alright, that was 1 and 2 proposed by Sleeden and Swan. I hope that's how they pronounce this name. I know this is pronounced as Swan, but I'm not sure about that name. I'm hoping it's Sleeden. Alright, and then the third one was given by Virchow, who said all cells arise from pre-existing cells. Alright, so sometimes I just reiterate whatever was given here, because theories are supposed to be written how they are. I mean, there is scope for change. Most of the time I do change it. I'm not saying I never change it, and I don't say I always change it, but sometimes I do, sometimes I don't. The times that I do, my teachers told me that this is the way it has to be written, so I memorized it. So even if I try, I keep coming back to this same version that was already mentioned in the textbook, alright? Okay, so that's an indication that, okay, this is important, this has to be written how it is, alright? Okay, so there are a few unicellular organisms like amoeba, chlamydomonas, paramecium, and bacteria. So one trick that I use to remember unicellular organisms is that at this level there are only 4. There will be more, but in 9th grade there were only 4 for us. There are only 4 even now. So the first one I remember is amoeba, amoeba. And the second one I remember is bacteria. The third one is chlamydomonas, and then the fourth one would be paramecium. Alright, so if you observe the first letters of all 4 of these, let me switch colors. It's A, B, C, P. Alright, A, B, C, P, like A, B, C, D, but the D is not D, it's P. So that's how I remember it. Amoeba, bacteria, chlamydomonas, paramecium. That's how I remember unicellular organisms. That's how my teacher taught me. Hope that helps you remember the 4. So whenever a question in the exam is like, name 3 unicellular organisms, you can just be like, oh, ABC, amoeba, bacteria, chlamydomonas, or paramecium, whatever comes to your mind. But just make sure that you remember the 4 of these, because there's a chance for messing up in giving names of multicellular organisms. That's a very sad way of losing points, in my opinion. So make sure you remember this ABCP, and I hope it helps you. Alright, moving on. Many cells group together in a single body and assume different functions in it to form various body parts in multicellular organisms. So in unicellular organisms, we saw that there is only one cell that live on their own, right? In that cell there are different organelles. That's a new vocabulary term. organelles that we will talk about later in this lesson, but I'm just going to go ahead of myself and mention it right here. Okay, so in unicellular organisms, there is a single cell that can live on its own, but that cell has many organelles that perform the various functions of a cell. In multicellular organisms, the organism itself is made up of various groups of cells, and each group of cell has a unique function. And in that group of cells, each cell has organelles again, and those organelles perform the different functions of the cell. Alright, so this is an important distinction that has to be made. Alright, so every multicellular organism has come from a single cell. And how is that? We know that cells divide to produce cells of their own kind, the third postulate of the cell theory, and all cells thus come from pre-existing cells. Alright, so we have an activity here that also has to be done using a microscope. And then we have a few cells. We have the diagrams of a few cells from the human body. This is also important. You need to know how to draw these diagrams. We have smooth muscle cells, blood cells, nerve cells, fat cell, bone cells, sperm, and ovum. You need to know how to draw all of these, and you need to know, you need to label them with what they are. All right, and then we move on to the third page. Soon I'm going to forget count, so I'm just gonna say next page. I'm not sure if this is third page also. Alright, so the shape and cell of cells are related to the specific function that they perform. Some cells like amoeba have changing shapes, and in some cases the cell shape could be more or less fixed and peculiar for a particular type of cell. So all this boils down to one thing. First of all, I want you to take the example of a neuron. Whenever you say neuron, the 90% of the time, I think whenever you say neuron, 100% of the time, the first thing that comes to my mind is this: a star-shaped cell body of sorts with branches here. This is a very rough diagram. I can do much better, I promise. Alright, so these are the branch-like structures, and this is like an axon, and these are the terminals. So whenever you say neuron, this is the first thing that comes to mind with the nucleus here. So when you say nucleus, this is what comes to mind, not that, right? This is not what comes to mind when you say neuron. Alright, so that is the meaning of this statement. In some cases, the cell shape could be more or less fixed and peculiar for a particular type of cell, right? And not all neurons look like this. There are a few neurons that look just like that. Alright, there's just one branch, and then this is the axon again. Okay, but they have a more or less fixed shape. There's a cell body, and then there's the axon, and then there's the terminal fibers and everything. That's the fixed shape that they're talking about here. So the first statement says the shape and size of cells are related to the specific functions that they perform. So what is the function of neuron? I'm going ahead of myself, but the function of a neuron is to transmit messages, right? So from one neuron to another, it should receive a message from this end and pass it on to the next end. So the most helpful structure would be to have a long and elongated structure. That's why all neurons are like this, whether they have 4 branches or 5 branches or just 1 branch. They're almost always very long. Alright, and some cells like amoeba have changing shapes. Alright, and that is also for a specific function, because it has to take in food, and it can only take in food when it keeps changing its shape, right? That is the whole meaning of this part of the lesson, and it's very important to understand the difference between and to understand the significance of the different shapes of cells. Alright. Each living cell has the capacity to perform certain basic functions that are characteristic of all living forms. And how does a cell perform these basic functions? We know that there is a division of labor in multicellular organisms, so that was what I was talking about. There are specific groups of cells that each have a unique function. So there are bone cells that form bones, which gives us the structure to the body. There are neurons that form nerves that transmit messages. There are other cells in the stomach that are useful for digestion. There are a few in the lungs for respiration. So that's all division of labor. There is not only one cell that takes care of all digestion, respiration, messaging. And there's not just one cell, right? There are many cells, there are many groups of cells. That is the division of labor that they're talking about here. So this means that different parts of the human body perform different functions. The human body has a heart to pump blood, a stomach to digest food, and so on. Similarly, division of labor is also seen within a single cell. In fact, each such cell has got certain specific components within it known as cell organelles. That's what I told you before up there. Organelles, each cell has organelles, and each group of cells have a cell which in turn has many organelles within that one cell. Alright. So each kind of cell organelle performs a specific function, such as making new material in the cell, clearing up the waste material, and so on. There are many functions there are many organelles with many functions that we will cover later in the lesson. Alright. A cell is able to live and perform all its functions because of these organelles. And these organelles together constitute the basic unit called the cell. So, organism is made up of many cells, and cell is made up of many cell organelles. Okay? So, in between here, there are many other subdivisions, but it's just a basic overview. There are tissues, organs, organ systems, all of that. But this is just a basic overview. Many cell organelles make up a cell. Many cells make up an organism. Alright. So it is interesting that all cells are found to have same organelles, no matter what their function is or what organism they're found in. Alright. And then we have a couple of questions that I would like you to try on your own. We covered both of these previously in the lesson, so if you're not able to answer them, do not worry at all. Go back to wherever I explained these, wherever you see in the textbook that I go back where I explained that part of the chapter. Listen again, make sure you understand, take notes, come back, answer this, and you will be ready to move on. Alright, so what is a cell made up of? And what is the structural organization of a cell? Alright. We saw above that the cell has special components called organelles. How is the cell organized? If we study a cell under a microscope, we will come across three features in almost every cell: plasma membrane, nucleus, and cytoplasm. All activities inside the cell and interactions of the cell with its environment are possible due to these three features. Alright, let's see how. First we're talking about the plasma membrane. This is the outermost covering of the cell that separates the contents of the cell from its external environments. Alright, so here I would like to make a side note that says the plasma membrane, I'm going to write PM, or the cell membrane. CM is the outermost membrane, is the outermost covering for animals only. And by animals, I mean humans as well. Okay, animals is anything that's not plants that have. So if you want to go deep, I can't give you every exception, but animals basically covers humans as well, not just a cat or a dog or anything. Okay, animals including humans, the cell membrane is the outermost covering. For plants, the outermost covering outermost covering would be the cell wall. Alright, the cell wall is the outermost covering for plants, and the cell membrane is the outermost covering for animals. Alright, the plasma membrane allows or permits the entry and exit of some materials in and out of the cell. This is also why the cell membrane or the plasma membrane is called a semi-permeable membrane. Okay, it's called semi-permeable or selectively permeable membrane. I think this textbook prefers selectively permeable membrane, so maybe your teachers also prefer selectively membrane, so I want you guys also to prefer selectively permeable membrane. Alright, so therefore it's called, yes, see, therefore it's called selectively permeable membrane. So how does the movement of substances take place into the cell, and how do the substances move out of the cell? Some substances like carbon dioxide or oxygen can move across the cell membrane by a process called diffusion, and I believe in the next chapter or later in this chapter there are a few more categories like diffusion that we will study about, but not to worry, we will take one step at a time. Alright, so we have studied the process of diffusion in earlier chapters. Hint chemistry. We saw that there is spontaneous movement of a substance from a region of high concentration to a region of low concentration. That is the basic definition of diffusion. Diffusion is the spontaneous movement. Spontaneous is the important key term here. Spontaneous movement of a substance from a region of high concentration, also important, to a region of low concentration, also important one, right? Low concentration. So spontaneous movement, high concentration, low concentration. Those are the three terms in order that you have to do in the rest of the sentence. As long as it doesn't change the meaning of the definition, you're fine changing it. Or that's at least how it was when I was studying. Alright, so something similar to this happens in cells when, for example, some substance like carbon dioxide accumulates in high concentrations inside the cell, and they have to be discarded from the cell because that's like the waste material. So it moves from high concentration, which is inside the cell, high concentration, to low concentration, which is outside the cell, through a process called diffusion. That is how cells get rid of carbon dioxide from their cells, through the process of diffusion. Alright, and then similarly, oxygen enters the cell by the process of diffusion when the level of concentration of oxygen inside the cell decreases. Alright, so again this is when the concentration inside the cell decreases. That's when it enters the cell, because in the cell there is low concentration and outside the cell there would be higher concentration. So from higher concentration, which is outside the cell, to lower concentration, which is inside the cell, oxygen diffuses. Alright, water also obeys the law of diffusion, and the movement of water molecules through such a selectively permeable membrane is called osmosis. Alright, again, osmosis, the basic definition is the movement of water molecules. Just going to mention the key terms. Water molecules, the movement of water molecules from a region of high concentration to a region of low concentration through a selectively permeable membrane. Alright, these are the four important terms. Alright, very important, because if you don't add these clauses in, that could lead to people assuming it to be a different definition, and that could lead to you losing your points in your exam, and then you're going to say, oh, I was looking at this YouTube video, but then, hey, it's not my fault. I mention that these four terms are important. Alright? Okay. So the movement of water across the plasma membrane, also known as the cell membrane, is affected by the amount of substance dissolved in water. So the concentration, that's what we're talking about here. So osmosis is the passage of water from a region of high concentration through a selectively permeable membrane to a region of lower water concentration until equilibrium is reached. Okay, so there's a fifth one, until equilibrium is reached, because it can't keep happening forever. That's what it takes place until equilibrium is reached. So what will happen if we put an animal cell or a plant cell, for that matter, into a solution of sugar or salt in water? So salt or sugar, water, what's going to happen if we put salt water? So one of three things is going to happen here. Okay, so let me clear this. Alright, so there's three situations. Situation 1, okay, situation 1 would be hypotonic. I'm just going to name them. I will explain what's what later once I name all of them. So 3 I'm just going to do here, hypertonic. Okay, so the three types are hypotonic, isotonic, and hypertonic. Hypotonic, let's just draw a cell here, and this would be the so this is the salt or sugar water. So a cell, if you don't have microscopes, you can also use sugar water with a raisin or a dried grape that's put inside it. Okay, so that will also give you the same results, typically the same results, but it's just on a larger scale, so you can even observe it better. Alright, so I'm just going to do three situations, and that's how it is. Okay, so hypotonic solution. If the medium surrounding the cell has a higher water concentration than the cell, meaning that the outside solution is very dilute, the cell will gain water by osmosis, and such a solution is known as a hypotonic solution. So the outside solution is very dilute. That means there is a higher water concentration in the medium surrounding the cell than the inside of the cell. Alright, so if we're another way to put it, if this is confusing you, is that higher water concentration outside means that there is lower water concentration inside, right? Why is that? Because there's higher particle or whatever else that we want to do. Because I said raisin in sugar water because raisins are mostly sugary foods, right? So they have higher concentration of solute. Keep this clear in mind. The inside of the cell has a higher concentration of the solute than the outside of the cell, which means that the outside of the cell has a higher concentration of the solvent than the inside of the cell. These are two ways that the question can be posed. So I'm just making it clear, and I'm stressing that in a hypotonic solution, the inside of the cell has a higher solute concentration than the outside of the cell, and the outside of the cell has a higher solvent concentration than the inside of the cell. Alright, and in this case, water moves in through the selectively permeable membrane through a process known as endosmosis. Alright, the movement of water from outside the cell to inside the cell is known as endosmosis. And isotonic endosmosis. And then we have so endosmosis is the movement of water inside the cell in the case of a hypotonic solution, Isotonic solution is a case in which there is equal concentration of solute and solvent both inside and outside the cell. Okay, so isotonic solution, it's the equilibrium point. So in the definition of osmosis, we saw that the movement occurs until equilibrium is reached, while the isotonic solution already has equilibrium reached, so there is no movement. All right, no movement. And then we move on to hypertonic solution, where if the medium has a lower concentration of water than the cell, it is a very concentrated solution, right? So if we have water here, all right. So hypertonic solution is a solution in which there is a lower concentration of solute outside the cell than inside the cell, or there is a lower concentration of, I'm not sure what I just said. There is a higher concentration of, yeah, right. There is a higher concentration of solute outside the cell than inside the cell, or there is a higher concentration of solvent inside the cell than outside the cell. All right, that is hypertonic solution, and in such a case, water moves out of the cell, okay, or the solvent moves out of the cell through a process called exosmosis. So endosmosis, okay. So endosmosis is a process in which water goes inside, endo means inside, and exosmosis is a process in which the solvent leaves the cell, it moves outside. Exo means outside, endo means inside. Alright. So that's one way of remembering it. Just like endothermic, exothermic reactions, endosmosis and exosmosis. Okay. So moving on. So what in all three conditions, the water crosses the cell membrane in both directions, but determining whether more water leaves the surrounding medium or whether it leaves the cell more, it depends on the concentration of water. Even in isotonic, maybe there is movement of water, like I'm just showing a hypothetical 3 arrows, but then there's also 3 equally there is an equal 3 solvent movement from inside the cell, so it just gets balanced and gets cancelled out. And in this case, there are less water molecules moving inside than outside, so the net movement is outside. In this case, there is also outside movement, but the net movement is inside the cell. Alright, so you can also try it with dried raisins or apricots if you don't have an eggshell or a cell. Alright, unicellular freshwater organisms and most plants tend to gain water through osmosis because they have a lot of water in their surroundings. So it's mostly endosmosis. And absorption of water by plant roots is also an example of osmosis. So when we water plants, we increase the concentration of water or solvent in the medium surrounding the cell. That is, we create a hypotonic solution situation. So the roots take in water through a process called endosmosis. I'm adding in extra points for NTSE and NEET preparing students, because that's going to help you understand concepts a little better later. And even for CBSE, you will find this helpful later. Alright, so plant roots absorbing water through the process endosmosis is because we create a hypotonic solution situation by watering the plants. Alright, this is why diffusion is important in exchange of gases and water in the life of a cell. And in addition to this, the cell also obtains nutrition from its environment. So the potassium, phosphorus, and nitrogen, the NPK nutrients the, I guess, macronutrients that the plant absorbs from its environment. That is also because of osmosis. And different molecules move in and out of the cell through a type of transport requiring the use of energy. So we studied diffusion as the spontaneous movement, right? And spontaneous movement is diffusion, and osmosis is a part of diffusion. So osmosis is also a spontaneous movement. That means spontaneous just means that it doesn't need extra energy to move. The particles or molecules don't need additional energy to move from a region of higher concentration to a region of lower concentration. Now, these types of transport that require the use of energy are known as non-spontaneous reactions. You might also find a term familiar, active transport. Okay, so active transport has other so just like how this is called active transport, this is called passive transport. And while passive transport includes processes like diffusion and osmosis, active transport you might find bulk movement or ion pump, words like that familiar. Those preparing for NTSE or NEET, you might find that familiar. If not, just Google it and see if that's going to help your preparation improve a little bit. Okay, and then we have two more questions here that we did cover previously. So if you didn't understand, again, just go back and learn. Make sure you understand, take notes, come back, answer, and then say, yeah, I'm ready to move on. All right, and then we're talking about the cell wall here. So the first of the three factors that are common in every cell, the first one was the cell membrane. This is the cell wall that I mentioned. The outermost covering in plant cells, that is before the cell membrane. So cell wall is a rigid outer covering which lies outside the plasma membrane. It is mainly made up of cellulose in plants, and it's a complex substance that provides structural strength to plants. Alright, that's the function of cell wall. It provides strength also. Alright, so when a living plant cell loses water through osmosis, this is exosmosis. I need you to keep saying it out loud, like whenever you come across terms like this. Alright, so that will help you remember better. Exosmosis, that's what they're talking about here. There is a shrinkage because when you imagine a plastic bag which is elastic, and you have say 10 apples in it, and all 10 apples were fit in that bag. As you keep removing apples from inside the bag might shrink because there are less contents in the bag. It will shrink, and that is because of exosmosis, because you're taking out apples from inside the bag to outside the bag, maybe from a region of higher concentration onto a table where there are no apples. If there are 10 apples in the bag and there are 0 apples on the table, that's supposed to be 10. Alright, if there are 0 apples on the table, this is a region of higher concentration, this is lower concentration. So as you remove apples, the bag will shrink, right? That's how cells shrink when they have exosmosis undergone inside them. Okay, alright. This phenomenon is known as plasmolysis. When the when the contents of the cell shrink away from the cell wall. Alright, so if this is the bag, there are fewer apples, so they might tend to get concentrated in the center or away from the sides of the bag. Alright, I mean this is not true for the example that I gave you, because apples tend to stick to the walls of the cover, but this is what happens in plant cells. The contents of the cell, if they were like previously like so, they now move to away from the cell wall. This is a rough draft of what you can see in your textbook for the diagram of plasmolysis, but that's the actual that's the concept of plasmolysis. So it's the definition would be the shrinkage or contraction of the contents of cell away from the cell wall is known as plasmolysis. And if you observe, I just reiterate and I just repeated what was given in the textbook, because that was one of the definitions that I had to memorize, because my teacher said it was important that I needed to write the exact definition or write loose points. Alright, so keep that in mind, maybe just like memorize that definition. Alright, cell walls permit the cells of plants, fungi and bacteria to withstand very dilute hypotonic external media without bursting. So when it's dilute, when the external medium is dilute, there is a tendency for the cells to take up the water to make the outside surroundings concentrated. And if you take up more than what you can hold, obviously you're going to burst, just like a cover that can take a maximum of 10 apples. If you put the 11th apple, it will tear, right? Just like how the bag tears, the cell will burst. But the cell wall in plants, as contrary to animal cells, plant cells have a cell wall which withstand that pressure and prevent the cell from bursting in when they undergo too much endosmosis, when they take in too much contents. Alright, this builds up a pressure against the cell wall, and the cell wall exerts an equal pressure against the swollen cell. Alright, and because of their walls, such cells can withstand much greater changes in the surrounding medium. So because they don't easily burst or like tear for covers. They don't easily burst when there is a difference in the surrounding medium, but animal cells are more prone to do that easily because they don't have that protective cell wall. And then we have nucleus. So remember that amount of onion peel that we prepared? We put iodine solution in the peel. That is, I didn't go through the activity, but if you go through, there was a step that said put iodine solution. That would be because we want to see the nucleus. Alright, so alright, so that is just an introduction of the nucleus, and here we have the actual content. So the nucleus has a double layered covering called the nuclear membrane, and it has pores which allow the transfer of material from inside the nucleus to its outside, that is to the cytoplasm. Those are called nuclear pores. Okay, so if this is a cell, if this is a nucleus, we're going to talk about that region in specific now. So it has two membranes called the nuclear membranes. Let me make it kind of neat. I'm trying to. This is the best I can do without wasting anyone's time. Okay, so in that there are a few pores. So one clever thing that I think would be, is to shade it like so. So these are the pores in between the nuclear membranes. Just assume these are pores and not random lines. Okay, so at one glance, if you just look at it suddenly, you might feel like there are a few gaps in these two circles, and these circles are called nuclear membranes. And these gaps will be called the nuclear pores. And through these nuclear pores, if this is the cytoplasm that's outside, cytoplasm again is the fluid, semi-fluid substance that has all the cell organelles present in it. Okay, so the movement of materials from the cytoplasm to the inside of the nucleus occurs through these nuclear pores. Alright, okay. Okay, so I'm just gonna get rid of this because I want to read what's here. Alright, so it contains, okay, so the nucleus contains chromosomes which are visible as rod-shaped structures only when the cell is about to divide. Okay, so chromosomes are visible only when the cell is about to divide. They contain information for the inheritance of characters from parents to the next generation in the form of DNA, and DNA is deoxyribonucleic acid. So whenever someone says, oh, you look just like your dad, or you look just like your mom, or your grandmother or grandfather, whatever, that is because of the chromosomes, and that is because of the information in the chromosomes that we look like our parents or our grandparents, or sometimes our aunts and uncles, because of the information, the similar information that's present in the chromosomes, and the chromosomes are seen only when the cell is about to divide. So what about what happens to the chromosomes when the cell is not dividing? Well, that is present as chromatin material. It is visible as an entangled mass of thread-like structures, and when the cell is about to divide, the chromatin material gets organized or gets condensed into chromosomes. Alright, so when the cell is not dividing, chromosomes are present as chromatin material, the thread-like, fine thread-like structures that are, they're just like one of the easiest drawings for me. Chromatin material, I can just do that, and that will be a perfect representation of the chromatin material. Alright, so the nucleus plays a central role in cellular reproduction because that's where the chromosome is, that's where the chromatin material is, that it plays a central role in a reproduction, in cellular reproduction, and because the cell is the basic unit of the organism, the nucleus plays a central role in the reproduction of the organism in itself. Okay, so reproduction is the process by which a single cell divides into two new cells. It determines the way the cell will develop and what form it will exhibit at maturity by directing chemical activities of the cell. Right? In some organisms like bacteria, the nuclear region of the cell may be poorly defined due to the absence of a nuclear membrane. Such an undefined nuclear region containing only nucleic acids is called a nucleoid. It's mostly seen in prokaryotic organisms. I'm probably going ahead of myself, but I need you to know this. Prokaryotic means primitive, okay? Pro means primitive and karyotic means nucleus. So prokaryotic organisms are organisms with a primitive nucleus, which contain just the nucleic acid called as a nucleoid. Alright, those are prokaryotic organisms. Such organisms whose cells lack a nuclear membrane are called prokaryotes. Oh, they covered it in the next sentence. Okay, prokaryotic organisms and organisms with a nuclear membrane and nucleic acids are called eukaryotes. Eukaryotes means a well-defined nucleus. Alright, prokaryotic organisms also lack most of the other cytoplasmic organelles that are present in eukaryotic cells. Many functions of such cells are also performed by poorly organized parts of the cytoplasm. Alright, they are seen in membranous vesicles, chlorophyll, but not within the plastids as seen in eukaryotic cells. Then we talk about the cytoplasm. Is the fluid content inside the plasma membrane. It also contains specialized cell organs. So all the cell organs are present in the cytoplasm, and each organelle performs a specific function for the cell. Alright, cell organelles are enclosed by membranes called the they each have their own membranes. A few of them don't, but most of them do. In prokaryotes, besides the absence of a well-defined nuclear region, membrane-bound cell organelles are also present. Alright, eukaryotic cells have nuclear membrane and membrane-enclosed organelles, and the significance of membranes can be illustrated with the example of viruses, which lack any membranes and hence do not show any characteristics of life until they enter a living body and use the cell machinery of the host to multiply. That's what the living body is called. It's called a host. And there is one more question here that I want you to try on your own. And then we talk about the cell organelles. Alright. Every cell has a membrane around it to keep its own content separate from the external environment. Large and complex cells, including cells from multicellular organisms, need a lot of chemical activities to support their complicated structure and function. To keep these activities separate from each other, the cells use membrane-bound organelles within themselves, and this is most common for eukaryotic cells, where they have well-defined nuclear membranes and membrane-bound organelles. Alright, so this is one of the features of eukaryotic cells that distinguish eukaryotic cells from prokaryotic cells. Alright, so now we have a few organelles that we will talk about. The first one is the endoplasmic reticulum, famously abbreviated as the ER. When I first saw it, I thought it was the emergency room, but that's just a funny joke. Okay, so the endoplasmic reticulum is a large network of membrane-bound tubes and sheets. Tubes and sheets, membrane-bound, large network. Important keywords. Alright, large network, membrane-bound, tubes and sheets. So membrane-bound is important because it shows that the endoplasmic reticulum is one of the membrane-bound organelles of a cell. Alright, so it is similar in structure to the plasma membrane, and there are two types of ER: RER, which is the rough ER, and SER, smooth endoplasmic reticulum. And RER looks rough under a microscope because it has particles called ribosomes that are attached to its surface. So if this is ER, let's say, there are ribosomes attached to a part of the ER that make it look rough. So it gives it this texture. So when you distinguish this part, let me switch colors, this part of the endoplasmic reticulum with this part of the endoplasmic reticulum, this the upper part shows a little texture and it's different from the second the lower half, because this shows a little structure here, and that is because of the presence of ribosomes. These dots that I drew here are the ribosomes. That's what gives the rough endoplasmic reticulum its rough look. Alright, so ribosomes are used for protein manufacture, and the manufactured proteins are then sent to various places depending on the need using the ER. That will be the second the second organelle that we will learn about. And then so rough ER has ribosomes which manufacture protein. So protein manufacture, that is, so protein manufacture indirectly is a function of the rough ER. Alright, and the smooth ER helps in the manufacture of fat molecules or lipids. That is important for cell function. And so the rough ER and smooth ER together help in a process called membrane biogenesis, which is the formation of cell membrane. Alright, and this is an animal cell. This is a pretty important, this is a pretty important diagram that you need to know how to draw for subjective tests and label for subjective and objective tests. Alright, so ER also serves as a transportation system for the between the various regions of the cytoplasm, or between the cytoplasm and the nucleus. This happens through the nuclear pores. So one way to remember what you studied is to constantly keep revising whenever you come across sentences like this. Like here, you had two options. You could either just read away and move on, or you could make a connection and say, oh, this is what we learned previously, these are the nuclear pores. That will help you remember better. Alright, this is the diagram of a plant cell. Alright, so the smooth endoplasmic reticulum also plays a crucial role in detoxifying many poisons and drugs from the cell. Alright, and then we have the second organelle called the Golgi apparatus, which consists of a system of membrane-bound vesicles or the flattened sacs. You could substitute vesicles for flattened sacs and vice versa. That are arranged approximately parallel to each other in stacks called cisterns. So whatever I underlined are important points in that definition. So these membranes have connections with the membranes of the endoplasmic reticulum that constitute another portion of a complex cellular membrane system. So when we were doing endoplasmic reticulum, I said the protein manufacture, the proteins that are manufactured by the rough endoplasmic reticulum are transported, which leads us to the second organelle. This was what I was talking about, the Golgi apparatus. This is involved in the second step of protein manufacture, which takes the proteins from the endoplasmic reticulum and transports it to the different parts of the cytoplasm. And then we have so the material synthesized near the ER, which is proteins. See, this is what I mean by connections. You need to keep making connections constantly if you want to remember what you learned. Alright, it is dispatched to various targets inside and outside the cell through the Golgi apparatus. Its function includes the storage, modification, and packaging of products in vesicles. In some cases, complex sugars may be made from simple sugars in the Golgi apparatus. It's also involved in the formation of lysosomes. That is what the formation of lysosomes is one of the more important, not most important, but more important functions of the Golgi apparatus. And lysosomes are membrane-bound sacs filled with digestive enzymes. So membrane-bound is important, and digestive enzymes is important because the main function of lysosomes is because of its digestive enzymes. So these enzymes are made up of they're made by the rough endoplasmic reticulum, and they are a kind of waste disposal system for the cell, which help to keep the cell clean by digesting any foreign material as well as worn out cell organelles. Foreign materials entering the cell as well as old organelles end up in lysosomes, which break complex substances into simpler substances, and sometimes lysosomes also burst and digest the enzymes in their own cell. That's why they're also known as the suicide bags, because they kill themselves for the to protect the cell, and because they kill themselves, they're known as the suicide bags. And then we have mitochondria, known as the powerhouses of the cell. This is probably the most famous cell organelle. Mitochondria, also known as powerhouse of the cell. They have two membranes. They're double membranous. Double membrane. Alright. The outer membrane is porous while the inner membrane is deeply folded. The folds increase the surface area for ATP generating chemical reactions, and ATP is a form of energy. So it's like a molecule of energy. ATP, that is adenosine tri means 3 phosphate. Alright, ATP here is also known as the energy currency of the cell, and the body uses energy stored in the ATP for making new chemical compounds and for mechanical work. And they mitochondria, let me switch colors here. Mitochondria And again, I'm getting a little ahead of myself, very little, but I am getting ahead of myself when I say, and plastids, they both have double membrane. Oh, no, they don't both have double membrane. They both have, if this wants to work, okay. They both have their own circular DNA. Own circular DNA. Own is important here. Own circular DNA and 70S ribosomes. So I'm just going to mention 70S. This is also important. And ribosomes are basically of two types, 80S and 70S. 70S is the lower unit, but mitochondria and plastids both have their own circular DNA and the 70S type ribosomes, which makes them semi-autonomous organelles, alright, because they have important, like the most important organelles in the cell, that they have their own DNA, and it makes them semi-autonomous. They're like half or autonomous. They don't have everything that a complete cell has, but they do have important parts. That's why they're called semi-autonomous. And plastids are only present in plant cells. We don't see them in animal cells. There are two types of plastids. Chromoplasts, colored, and leucoplasts that are white or colorless. Chromoplasts that have chlorophyll in them are called chloroplasts. Chloroplasts we are very aware of because that they contain chlorophyll, and chlorophyll we see in most green plants. I mean all green plants, which are most plants. Alright. Chloroplasts also contain various orange or yellow pigments in addition to chlorophyll, but it's just that the green chlorophyll pigment is dominant in them. Alright, leucoplasts are family organelles in which materials such as starch, oils, and protein granules are stored. Later, I guess later in the in the course, or maybe in 10th grade NCERT, we learned about the different names for starch, oil, and protein. Granules, the plastids, amyloplasts, and there's 3 different names for starch, oils, and proteins that we don't need to touch now. The internal organization of the chloroplast consists of numerous membrane layers called the stroma. When I first read this, I thought it was just a typo with the R, but the R is there for a reason. Stroma is the membrane layers that are embedded in the that's the fluid material that is present in the chloroplast, while stoma is the singular form of stomata, and the stomata are the tiny holes that are present on the underside of the leaves for respiration. I think we learned about them in 8th grade or 7th grade, just before 9th grade. Alright, and then like mitochondria, they also have their own DNA and ribosomes. And then we're talking about vacuoles. They're storage sacs for solid or liquid contents. Vacuoles are small sized in animal cells, while they're very large in plant cells. The central vacuoles are very large in plant cells, which contain from about half to almost the entire cell. So 50% to 90% is like half to almost the entire cell in plant cells only. In animal cells, they're very small. Sometimes they're not even present. Alright. Vacuoles are full of cells in plant cells, and they provide turgidity and rigidity to the cell. Alright. And many substances of importance in the life of the plant cell are stored in vacuoles. So vacuoles are the storage units. They store almost everything, all the important stuff. Alright. So amoeba has a food vacuole that takes in all and stores all the food that that amoeba took in. And in some unicellular organisms, specialized vacuoles also play important roles in expelling excess water and some wastes from the cell. And we have 4 questions. We did all of them. If you didn't understand them, go back to where I explained them. Make sure you understand them, come back and answer them, and then let's move on. Alright, so each cell acquires its structure and ability to function because of the organization of the organelles and its membrane. And this is why the cell is the fundamental structural unit of living organisms. It's also the basic functional unit of all life. And then we have cell division. This is the last concept for this chapter. So for those of you who were getting restless, rejoice. Alright, so new cells are formed in organisms in order to grow, to replace old, dead and injured cells, and to form gametes required for reproduction. So it covers 2 functions that were explained in 2 halves of a sentence. Whenever you fall down, if you are hurt, or if you have a layer of your skin peeled away, maybe in a few days or a few weeks, or sometimes few months, your skin gets healed. It forms that outermost surface again. How does that happen? It's not like we pasted on a part of someone's skin or our own skin on there. Then how come there is newer skin that just miraculously appeared there, right? That is through cell division that skin heals when you have cuts or wounds or scrapes. That's how we heal, through cell division. And for reproduction is also because of cell divisions. That is how a single cell for prokaryotic organisms like bacteria, single cell divides into 2 cells. In that case, the single cell was reproducing to form 2 new cells, right? That is also through cell division. And to form gametes in multicellular organisms, it forms gametes, and the gametes of 2 parents form a zygote. Again, I'm getting ahead of myself, but I just want you to get an idea, so when you hear these words again later in the course, or later in 10th grade or 11th grade, you can think back and say, oh, that's what we learned about in the first chapter of biology in 9th grade. So that will give you an idea that when you hear zygote again, the first thing that will come to your mind is cell division, right? That's what I want you to do, and that is why I keep going ahead of myself to give you guys an idea. Alright, so there are 2 main types of cell division, mitosis and meiosis. So mitosis is this diagram right here. This is important, and it's important because you can see the difference between mitosis and meiosis during by looking at these 2 diagrams here, right? In mitosis, it's just like 4 cells here. I mean, 4 chromosomes. They get divided into 8 single chromosomes. Alright, but in meiosis, they get divided they still get divided into 8 single chromosomes, but if you notice, there are different, they like get crossed over, they undergo the crossing over process. NEET students or NTSE students, I expect you to know what crossing over means. CBSE 9th grade, it's okay, you don't need to know it, but if you want to know it, by all means go ahead, Google it, and know extra information so that you can understand everything better. Alright, and then we have meiosis is used for reproduction, where a gamete is formed because of meiosis. Let's suppose we all know humans have 46 chromosomes. In that, the mother contributes 23 and the father contributes 23. And this happens because of meiosis. Now let's consider what happens without meiosis, alright? So let's say generation 1 humans had 46 chromosomes each. So the mother gives 46, the father gives 46, and the child has 92 chromosomes. So the second generation, the mother has 92, the father has 92, 184. The third generation, the mother has 184, the father has 184, and that gives 8, 6, 3, 368. It keeps increasing like that. And how much will you keep increasing? The chromosomes can't keep increasing like that, right? So that is how a 48 chromosome human has 23 chromosomes in the reproductive organs because of meiosis, thus maintaining that constant 46 chromosomes in every stage of reproduction in every generation. Alright, so that ends the lesson. We have a few recap points here and that you can go through on the day of your exam or the day before your exam. We have a few exercises that you can try once you feel confident, and if you have a doubt in any of the concept, feel free to go back and do it and just play the video again. Alright, so let me clear all this. Alright, so I hope I got everything. Oh well, okay, don't want to waste time. So first we saw the introduction of cells. We talked about a few experiments. We talked about how you do need adult supervision if you're going to use sharp objects, or in chemistry if we were using chemicals. I wanted you to do it under adult supervision. Alright, that's what we learned about. We learned about the introduction, and then we learned about cell organelles. I'm just doing the main things that we talked about. In that we talked about cell membrane, in which we talked about osmosis. We talked about endosmosis, which is used for hypotonic solutions. And we talked about exosmosis, which is used which is seen in hypertonic solutions. And we also talked about isotonic solutions, in which there is neither endosmosis nor exosmosis. We learned about cell wall, which is present only in plants, contrary to cell membrane, which is present in plants and animals. We learned about the nucleus, in which we learned about the nuclear membranes and the nuclear pores. And then we learned about cytoplasm, which is the fluid-like substance in the cell. We learned about smooth endoplasmic reticulum and rough endoplasmic reticulum, the presence of ribosomes on the rough endoplasmic reticulum that gives it the texture. And then we learned about Golgi apparatus, which transports the proteins that are manufactured by the ribosomes present on the rough endoplasmic reticulum. We talked about lysosomes, which are also known as the suicide bags. We learned about mitochondria, which are the powerhouses of cells. We learned about plastids, chloroplasts, chromoplasts, and leucoplasts. These two are known as semi-autonomous organelles. And then we learned about vacuoles, which are present in small sizes in animal cells and big sizes in plant cells. And then we learned about cell division. In it we learned about mitosis, which occurs in all general cells, and meiosis, I hope the spelling's right, it looks right, which is seen only in reproductive organs. Alright, and just like that, I just covered the whole video in 3 minutes, I guess, for the points to recap. So if you think I went fast, which I think I did too personally, I want you to go back, set the speed to 0.75 and make sure you got everything. Alright, that ends the chapter. I hope everyone understood, and I hope this video helped you understand the concept better. So feel free to head on to the next video, chapter 6 in NCERT Science for class 9, which is also a biology chapter that covers tissues. The next level after cells, tissues, that's chapter 6. And don't forget that you can do this. Have a great rest of your day. Don't forget to like and share this video, subscribe to our channel for more content, and rock your exam. All the best. Bye bye.